Okay. Okay. Welcome all to the second lecture of the 任國榮先生生命科學講座. This lecture series was established a few years ago and is endowed by the Mr. 任國榮Memorial Fund. Okay? So each year we invite eminent biologists, life scientists to come to New Asia College to share with us his views on certain aspects of life science. So this year we are very glad to have Professor Peter Ng with us. He is from the National University of Singapore. It is indeed my great honor and pleasure to introduce Professor Ng to you. He is a good friend of mine. He is known to be a very well-famous scientist working on crabs and fish as well. So as you know, each year we focus on one special theme on life science. And for those of you who know something about life science, one of the major crises we are facing is the so-called biodiversity crisis. Okay. So Peter will now share with you his views on conservation. Before his talks, I'd like to introduce Peter a little bit. Peter, I think, is very proud that he is solely trained in Singapore, right? So he got his bachelor and PhD degree from the National University of Singapore. And after that, he stayed there as a faculty member and now is a professor at the Department of Biological Science of the university. And he is also the founding head of the Li Guangqian Natural History Museum, which was established a few years ago. But other than that, Peter is involved in many different positions within Singapore and internationally. So I don't want to name all of them. They are actually in the introduction which most of you have with you. But Peter is, of course, a foremost freshwater field biologist. He has visited many different places in Southeast Asia, collecting crabs and fish and so on. And he is also known to be a very good fundraiser. The museum that he established, he has, and so on, he just, as I know, almost do it single-handedly to raise all the funds. And over the years, he has received numerous awards. To name a few, that includes the Singapore National Youth Award, the National Science Awards of Singapore, and also the Asian Young Science and Technologies Award. Okay, that is actually quite a few years back when he was young. And so I think I should not spend too much time. So I want to give more time to Peter to talk with you on his views on biodiversity conservation. Peter, please.
Thank you, Professor Chu. 唔該晒啦. Forgive me, I will talk in English because I'm more fluent in English. If I speak in Cantonese, you're going to have a hard time understanding me. So the topic I have chosen today is something very close to my heart. It's about the science of taxonomy. So it's about how we recognize and separate different kinds of animals on the planet. For me, of course, I like my crabs the most. It's an animal which I've spent 40 years of my life studying. So it's quite close to my heart. Before I start, of course, I have to thank, of course, the Chinese University of Hong Kong for inviting me to come down to speak to such a big audience. Right? Most of you, of course, I know are students doing all sorts of different disciplines, not just biology, not just science. So I will try to convince you what I'm talking about, biodiversity and taxonomy, is important. I also have some friends who have come from the other universities to come down to listen to me because they're very worried that I'll say something dangerous. And I can promise them I will. So let's get started. The first thing, of course, all of you will be asking is, what is this title about? Well, I chose this title to talk about taxonomy because this is Hong Kong, right? And this is a term which is very famous for people who like tau ma. If you're horse racing and you like to bet and make money from horses, you can make a lot of money if you do a trifecta. On all three horses, first, second, third, all coming in the order you predicted. Of course, the word trifecta started with racing and gambling. But over the last 10, 15 years, the English dictionary has used the word trifecta very often. Every time you have three things happening at the same time that people think can change a big event, they call it a trifecta. A trifecta of events that can change something. For me, this is something about the science I'm talking about, which is taxonomy. Of course, the science which I'm talking about, taxonomy, is how you recognize and separate different kinds of animals. It's a science which is very, very old. It first started in 1758 from a Swedish scientist called Linnaeus. All the scientific names we use today, the classification systems we have for all life on Earth, starts from Linnaeus in 1758. It's a science that goes back 200 over years. It's an old science. But it's a science because it's so old. A lot of people have different ideas of what taxonomy is supposed to do. So we will discuss that today. Now, when you are studying animals on Earth, one of the big problems we have is that there are actually a lot of species on Earth. For those of you who don't do biology, you might think there are a few thousands of species. Well, we have about almost 2 million plants and animals that have scientific names. So we know what to call them. Scientists give them a name. We think there could be 10, 20, 30 million animals out there that don't have names. And this is what I like to do. I like to go and find strange crabs and fish that people have not seen before. Animals that have no names, and it's for the scientists to find them. Now, if you look at the numbers, 2 million with names, 10 million without names, and this is a science that's 200 over years old, what it tells you is we have a lot of work to do. And there is a problem. Today, most young people, and I'm sure it's the same in Hong Kong as it's the same in Singapore, believe a lot in environmental issues. You believe in conservation. You don't want all the animals and plants in your country to die out. You want to make money, you want to live a good life, but you also want to keep your heritage. You don't want the animals living in your country to disappear. Well, to do that, we have to find out what they are. Unfortunately, because science is so big and there's so many things to do, there's a general feeling that the kind of people like me, taxonomists, are very few. And maybe we can't finish the job. There is a word we use. It's called the taxonomic impediment. That's the word up there. It means there are too many species and not enough scientists to find out what they are. And of course, we need names. Ecologists, conservation biologists, all sorts of scientists need to know what are the animals out there. And the complaint is we are too slow. So I'm going to share with you some of the ideas I have and some of the feelings I have about my science. After 200 over years, we are still very slow. There are too many animals. Let's look back in time. When I first started 30, 40 years ago, these are the things I used for my science. Some of you, which are older, will remember what they are. The younger ones, you probably are on your phone now finding out what is an aerogram. All right? What is a typewriter? In the old days, when you want to communicate with a scientist overseas, you write them a letter. You wait 2 weeks for their reply. No email. Anything you want comes through the mail. You can't make phone calls, too expensive. You want to fly, not so easy. Everything you do is slow and easy. You use a typewriter, you type wrongly, you got to erase it and type again. This was the kind of science we did in the old days. Well, not old days, just 30, 40 years ago. I like that world because it was a more peaceful world. Now with email, I think it's terrible. People send you an email and they say, hey, I sent you an email 30 seconds ago, how come you haven't replied? I'm in a lecture. Yeah, lecture also can send message. Well, this is the past. Now, we are living in a new century. This new century is giving us technology which is absolutely fantastic. All right? And all these new tools is what I'm going to talk about, this TRIFECTA thing I'm going to talk to you about. Okay? Because we have a lot of new technology that is supposed to help taxonomy, help science move faster. To help taxonomy and to help actually many sciences. What I call a trifecta of solutions that actually should make people like me move even faster. We have digital media, rapid publication, right? And of course, we have the power of genetics and next generation sequencing on our tables. And when I talk to young people doing genetics, what they tell me is everything is going faster and faster and faster and cheaper and cheaper and cheaper, which means it's easier and easier and easier. So therefore, people like me who discover and find new species should go faster as well. Well, this trifecta of solutions, unfortunately, doesn't make me go much faster, and I'll tell you why. Okay, you have digital communication. You know, I love digital communication. In the old days, when I go for an expedition, I go out in the jungles to catch animals, okay? I cannot carry all my books with me. Now, I can carry my laptop with a small disk drive, and I can have 1,000 papers inside there, 1,000 books, all PDF'd. I just came back from a 3-week cruise in the Indian Ocean, all right? We were catching crabs from 1,000 meters. On the boat, I had my computer, and every time a crab comes up, I can check. Ah, it's a new species. Ah, it's not. Ah, it's an old species. Because everything is at my fingertips. It's fantastic. Does it help? Yes. Does it make me more excited at the spot? Yes. Sounds good. I've got digital technology. You take a picture. In the old days, we used slides. Now, everything goes on a digital camera. With Photoshop, you can make the animal look beautiful. The left-hand side is a photograph you take when you are, in a fishing port. You see a nice crab, you take a picture. The light no good, no choice. Once I finish with Photoshop, it looks as if I'm a genius photographer. When I publish it, everybody says, Peter, your picture is very nice. I say, yeah. My Photoshop skills are very good. So, it's easier. It's faster. The drawings you make, the photographs you take are so much more beautiful. People reading it straight away know what you're talking about. I'm an artist with Photoshop. Not a real artist, but, not too bad. For the older scientists here, you remember, when you send a paper for publication, 10 years, 20 years ago, if they say, I will publish a paper in 1 year's time, you're very happy. Now, the young people, after 2 weeks, hey, still not out yet. We are all more impatient, partly because electronic media makes it so fast. You don't even need a hard copy. You don't even need printing. Everything is electronic, right? You can send a paper out today, and in a few days' time, it may be published if it's that fast. It can be that fast if you're that good and lucky. People now talk in terms of days, weeks, at most months. I used to remember 1 year, 2 years if you're lucky. The world has changed. So all these seems to be helpful. It does make things go very fast. DNA, the third trifecta, is a powerful tool. In the old days, we have to use morphology, what the animal looks like. Nowadays, with people doing DNA, next generation sequencing, and all sorts of wonderful things, we can get more data about the animals. Are they genetically related? Who are they related to? Used to be slow. Now it's fast. Used to be expensive. Now it's quite cheap. Is it good? Of course it is. I have more evidence. Tell me whether my animal is new or not new. With this technology like genetics, I can start mapping out evolutionary patterns, how animals are related, right? So like this is one of these evolutionary trees of crabs. I'm doing it with some of your staff, right? I mean, you see this is your tree, right? We are using genetics to map out evolutionary patterns of crabs. It tells us interesting patterns. Some of the patterns I never expected. I'm excited as a scientist. It gives me powerful tools. It helps my taxonomy, my science become stronger. With DNA, I can even do very quick environmental impact assessments. For example, if I go to a place, I want to find out roughly how many species are there, I can use rapid DNA methods. They are not precise, but they are fast. And they are pretty accurate to some degree. I can use all sorts of mass sequencing methods to tell me certain areas have more biodiversity or less biodiversity. There are people who are proposing you can do what you call turbo taxonomy, fast-track taxonomy. You can publish 30, 40 species at one shot because you have all this trifecta of equipment and technology at your hands. Taxonomists can go very fast. That's what they say, right? They call it turbo taxonomy, you know. Nowadays, everything turbo must be fast, okay? And the argument is, with all these technologies and tools at your disposal, surely you can describe this 10 million other species very fast. Should be easy, right? Well, these technologies are real. Do I use them? I use them, okay? As a taxonomist, however, I have a problem because I'm still slow. And people are complaining, why are you so useless? With all the technologies at your disposal, why can't you finish describing all the crabs? You know, every year now, we are still finding 30, 40, 50, 60 species of new crabs every year around the world. Not just me, right? Can't you guys do faster? Can't you do 2, 3, 4, 500 a year? Well, the problem is, we can't. And I'll tell you why. A paper written about 4 or 5 years ago by a colleague of mine, okay? He did a detailed statistical analysis of species discovered over the last few years, a number of scientists doing it. And he did a wonderful statistical analysis, and his conclusion is, mo manthai. Species, almost all found. Taxonomists, more than enough. Because you look at all the papers published, you put everything together, it's no problem. Well, I like statistics, but statistics don't always tell the truth. Science is one thing. Number of people actually doing the work is a separate thing. And now I'm going to discuss with you, is the taxonomic impediment real or not real? First thing, you know, whenever people tell you there is a wonderful solution, okay, there is something that's going to solve all your problems. If you meet a doctor that says, I can cure all your diseases, just like that, the first thing you will do, Hong Kong people, Singapore people, is San King Peng. All right? Not possible. Because the reality of science and taxonomy is, there is actually a lot of layers and things you need to consider. The history and what's actually happening on the ground. Now, I said the science is 200 years old. 200 over years old. Yes, it is. A science that is 200 over years old, dealing with names, means there is a lot of baggage. Names which were used by Mr. Linnaeus in 1758 are still valid today. Our species name, Homo sapiens, came from Linnaeus. We cannot change the name because it's in the rules. And that is the name all scientists use for our species. Now, that is deep history. Taxonomy is different from some sciences in which today you have a theory, tomorrow you change, you throw the theory away. The names will stay. Each of you, when you were born, were given a name. That name stays with you until you die. You may not like the name. Sorry, no choice. The law holds you to that name. 200 years of history, 200 years of scientists from around the world discovering animals means there's bound to be a lot of confusion. Some animals have been described more than once. If we look at the science from 250 years ago, you cannot compare to today. The technology and the things they have are different. If you look at a specimen, a dead body of an animal collected by Linnaeus, you can cry. Why? It's dried. It's broken. No color. Not much information. Half the time, you don't even know where they put it. You have to find it. Where is the specimen? You search the world. You'll be a detective. You cannot just say, 揾唔到，冇相干. You have to find it. Okay? So that takes you a lot of time. You cannot just say, don't care. You have to find it. Then you have to check it. Sometimes only one leg is left for a crab. From that one leg, you have to decide what the animal is. Not easy. Where did Linnaeus get his thing? Somewhere in Asia. In those days, sailors travel around the world. Not like today, so easy. 200 years of history. Now, in zoology, in taxonomy, there is a rule book. For those of you who study physics, chemistry, mathematics, the international scientists have a rule book that says you cannot do whatever you want. Chemicals have a name. It follows a set of rules. Animals and plants have also a set of rules. This group of people, the Commission for Zoological Nomenclature, they have a law. It's called a code. You cannot do whatever you want. You have to follow the scientific rules when you name animals. You have to follow the rules when there are problems. These rules are important. Same way as any law you have in every country, is to keep order. These are rules that make sure that every animal on this planet has a scientific name based on Latin. It has to be Latin. If it's not Latin, we have problems. Why? Because Latin was decided by Linnaeus in 1758, and I'm so happy he chose Latin. If he chose English, I guarantee you the French will protest, the Italians will protest, all right? And today in China, China will protest, okay? The Chinese will say, you know, we are 1 billion people. All names should be in Chinese. The Russians will demand difference. And Americans want it in American English. So, Latin is a dead language. It's a scholar's language. Just nice. The International Commission of Zoological Nomenclature is a group of about 25 to 30 senior scientists who meet to discuss whenever there are problems. When scientists are disagreeing on names, when they are fighting over names, they have to sit down like in a court of law to decide which name is correct or wrong. I used to be on a commission for 18 years, right? But after 18 years, it's time to retire to let young people do it. So I retired last year. It's an interesting commission because you have to deal with all sorts of strange problems. You know, names are very personal. It's like your own name. If somebody changes a name, you will go to court to fight them. Scientists do the same. They argue all the time. All right? And very often, the commission will have to vote to decide if the name is good or bad. The commission, like in all science, has rules. The rules basically say you have to respect the people you're doing. You cannot insult people. You cannot use stupid names. You cannot name the animals after somebody, not very nice. So the commission makes a judgment on names. Every year, the commission deals with 40, 50 problems, and they have to be solved. Now, all these legal things, all this history, means that any taxonomist identifying animals need to have a lot of history behind him. He needs to understand the whole history of a name. If you have a new animal, I find a new crab in Hong Kong, what do I have to do to be sure it's new? People always ask me, Peter, how do you know your crab is new? Because I checked through all the records for the last 250 years. Are there a lot of names? Yes. For crabs, we have 7,000 species of crabs today. How many names do I have to check? About 15,000. I need to know them to be sure my species is new. It's a lot of science work, it's a lot of detective work. Not easy, it's slow. We have another problem in taxonomy, and that is universities and museums actually already have a lot of dead bodies. People have been collecting animals for many, many, many years. If I go to any average biology department, even in Chinese University or Hong Kong U, I'm going to find behind their cupboards lots and lots of bottles of specimens. Museums got even more. With so many specimens, why do some of us still go out there and catch more? Why do we need to go around the world looking for new animals? Why did I spend 3 weeks out in the Indian Ocean chasing after crabs? Go to the museum easier. Well, not so simple. There are a lot of habitats and a lot of places that people have not been before. We were sampling at 1,000 meters. Very few people catch animals from 1,000 meters. It's not easy. It's not cheap. It's not fast. Not only are we getting new animals from new places, one of my most important jobs as a scientist, as a taxonomist, is to find 老朋友, old species. Species which Linnaeus found from somewhere in Asia. I want to find them where in Asia. I want to catch them now fresh, with color. I can take the tissues for DNA. I can use the animals for more work. Those old specimens, I can't use. And I need to know what they are. I need to find old, I need to find new. That's why we do expeditions. People think people like myself go out there always to look for new species. Actually, no. I like to find species people have never seen for 100, 200 years. They are more important. At the beginning of this talk, I said, is it possible that we actually have found most of the species? Is it that all the taxonomists around the world, they're more than enough to do the job? And if they're more than enough, why are we so slow to find more species? I've got some ecology friends. I always quarrel with my ecology friends. They'll go to a place, they will catch lots of crabs, for example. Put them on my table. Peter, tell me what they are. I say, all right, wait a few months. What? Can't you tell me next day? You're a crab expert, what? I say, yeah, but you have a lot of animals down there, got 10, 20, 30 species. Each one I need to check. I take a few days. So slow, all right? Sometimes it takes me a few weeks, a few months, a few years, and they always complain. You are destroying my science. You don't give me the information, I cannot publish my big paper. So they get very annoyed. They always say you're too slow. Now, we are slow because there are not enough of us, and there's a lot of work to do. If you look at a graph like this, it basically tells you the number of species been discovered is getting faster and faster, and there's still a lot of species. The upper graph is how fast we are finding species. The rate of discovery is still in the thousands a year. We are not slowing down. The number of species of different animals, insects, crabs, fish, are going up faster and faster every year. And with new technology like DNA, we are finding that it's even more. And all of them have to be named. one by one. Yes, it's a bigger playing field. But there is a problem in taxonomy that's very, very unusual. I said that in order to be a taxonomist, one of the things we have to do is we need to know the whole history of 200 over years. You need to know where the specimens are kept, where they are found, are they lost? For example, when I'm looking for specimens in Germany, what do I need to do? I need to find out what happened during the last 2 world wars. Was the town bombed? A few years ago, I was in Vietnam looking for some specimens collected by a Vietnamese scientist. We couldn't find some of those things until I found a very old scientist who told me, you know, Peter, when the Americans were doing bad things to Vietnam, like bombing us, we took the specimens, we hide them in the forest, and after that we can't find them. And it sounds very irresponsible, I thought, you know. You take specimens, put them in the forest, and then you lost them. But when people are throwing bombs on you, well, you're trying to be understanding. So where are they now? They don't know. Maybe eaten by some animal. I have to check all this, be sure that it's lost, and find fresh ones. Because I need replacements. The field is bigger. There are more scientists than ever before. But scientists are doing many, many things. Not every scientist can identify crabs or fish or insects. You need a specialist. And this is where, when I study taxonomy and some of the sciences you know, in every field, there are what we call big players. This is for crabs, okay? Crabs and decapods. There are 15,000 crabs, prawns, lobsters, crayfish. That's a group I'm most familiar with. There are about 7,000 crabs, okay? If you look at all crabs, prawns, and lobsters and so on, okay, 15,000. About 8,000 have been named only since 1950. That means half of them were discovered only in the last 50, 60 years. Not earlier. The old people from Linnaeus' time even slower. So we are actually doing a good job. Out of these 8,000 species, the scientists naming them is only about 1,500 scientists. Okay? What we call authors, people who write the paper. Now, here's the most surprising statistic. 30% of this were named by about 20 people. These people know the groups, know the animals so well, they can give names. Why? Well, they have seen the original specimens. They know where the things are. They have seen a lot of them, so they can tell you it's new, it's not new, it's old. So, 30%, 20 people. How many are alive today? 6. And 1 is standing here. 59% are by single scientists. And this is a problem in science that is very complicated. If you look at the papers published before 1950, most papers are written by 1 or 2 scientists, 1 or 2 authors. Today, the average number of authors on a paper is 3 or 4 or 5. What does it mean? It means that the modern people are lazier. You need more people to do one job. Does it mean that? Well, sometimes. What it means today is, whenever you discover a new species, when you write on a new species, there are other people also helping you. One person maybe is the one who did the ecology to help you catch the animal. One person who did the DNA work to help you understand it. More people are participating because there are more people involved in the science. Modern science, 21st century science, is more collaborative. More and more players come in. It's not because of lazy. It's not because of anything else, but more people are involved. But it doesn't mean that every person writing the paper is an expert on the animal. When I work with my colleagues on crabs, sometimes I put the name of the person who helped me find the animal. Why? Because that person almost died catching it. Is it fair that he's an author? Of course he is. Without him, I don't get my crab. Without him, I don't know what my crab is doing. Happy to have him. Somebody will tell me to do the DNA and tells me, you know, this one is a very funny one. Okay, also can. Science is collaborative. But for the crab, the only person who knows the crab best is actually me. How many taxonomists are there actually in that paper? 1. How many scientists? There can be 3 or 4. But the person who knows the animal, not that many. The statistical analysis I showed you just now has one big mistake. It adds everything up as the same. All the thousands of authors, most of them are not taxonomists. They are scientists in other disciplines helping. It's not the same. And that's where the problem comes. The problem is you need a group of people who have many, many years of experience with crabs. People always ask me, Peter, how come you know your crabs so well? Actually, I don't know why I know my crabs very well. I like them very much. Okay? And I've been studying them for 30, 40 years. When I first started, I liked crabs, so I studied them. Every year, I read more, I see more. The longer I live, the more experience I get. One reason why I know the crabs so well is because I spent 40 years chasing them. I've seen a lot. And every year I learn something new. When I read the papers I wrote in 1990s, I feel a bit embarrassed. Because when I read it, I say, wow, I shouldn't have said all these things because now I know so much more. But that's how science works. Step by step, you improve your knowledge because you see more things. You know, it's almost, when I say taxonomy, it's a bit different from the other sciences. Some sciences, you learn, you practice a lot, and you become very good. Some sciences is like the Chinese wuxia movie, the old masters. Right? The Shifu accumulates a ton of knowledge because he's lived long enough. Right? And this sort of information, I cannot just pass on to somebody. It's not possible. Knowledge can be passed. Experience and life experiences cannot be transferred. And in some disciplines like taxonomy, you need to be around for a long time. Now, I'm lucky to have been working on crabs for 30, 40 years. I know, I've seen a lot of things. My old Shifus have taught me a lot of things. They tell me things that are not in the books. I tell my students things that are not in the books. I talk to my colleagues working on crabs, and most of them ask me, how the hell do you know this, Peter? Because I happened to see this 10 years ago. Why? Well, because I happened to see it. Who told you this? Oh, this old professor who passed away 10 years ago. Taxonomy has a lot of history. The old teaches the young accumulates, and the information passes on. This is how I describe taxonomy. A bit of art, a bit of science, and you need a lot of time. Can you be a taxonomist, a good taxonomist, in 5 years? No. I've been doing this for 30 or 40 years. How do I rate myself? Ma-ma-de lah. I think I know a fair amount. I still got a few more years to go. I hope to live a bit longer. In the English language, for those of you who like philosophy and poetry, there's an expression that's called holding the center. It's also used in management. Okay? It's from a very famous poem by William Yeats. Holding the center means how do you ensure the science moves ahead when the core can be kept. One problem I have with taxonomy and modern biodiversity science is this. And this is almost the same as in our economy. What do I mean by economy? You know, one of the problems I talk to my friends from the business school is this. These days, in the modern economy, So many people are doing investments. The richest people are all speculating on stocks. What is missing? What is missing are the builders. The people who actually create products that allow your stock market to exist. It is a dangerous economy because if the people building things are fewer than the people playing the stock markets, sooner or later you die. The economy will collapse. You cannot speculate if you don't have anything to speculate. This is a worry of the economy. In science, in taxonomy, I have a similar problem. In my university, and I'm sure it's in your university, we have a lot of people now doing high-impact science on conservation. They take data of different kinds of animals and they do analysis on conservation, environmental issues, patterns, and they take this data and they crunch it together. All the high-impact papers on biodiversity, many of them are crunching data, data analytics. People like me, we generate the data. The problem? When you write a high-impact paper in Nature or Science, you are a genius. If you are the person describing the species, low-impact science. The people who create the data are less appreciated than the people manipulating the data. That annoys me. We must value the data creators because the data creators are the same people that are building industries, building companies. They start small. They don't make you big money, but they are the building blocks to the future of science. In taxonomy, you can't get rich because the names, the animals you find cannot be patented. There is no IP in a scientific name. There is no impact factor in a scientific name. It takes time. It's not easy to get famous. It's slow. The three tools, the trifecta of AIDS, are just tools. They help you do the job better. They can help you do the job faster, but they can't really move things that fast. At the end of the day, you still need those few people doing the hard work. And those few people are getting less and less every year. And that is a problem. Of course, somebody asked me, Peter, does it mean that taxonomists should die? One day, all the museum and university will kick them out. No more taxonomists. 绝种. No, actually, I'm not that cynical. Taxonomists are strange people. After listening to me for the last half an hour, you know that I'm not very normal. We like what we do. We don't really care if our bosses tell us this is not important because it has to be done. And taxonomists come from all circles. I've seen them from all around the world. The best ones don't really care how much reward they get. And in fact, that is the nature of science. You do the science because it's fun. You are answering questions that people don't know. Things that people have not found or seen before. It's like a wonderful hobby. It's great fun. And it's important. But taxonomists have to evolve. I'm a taxonomist for many years. Therefore, I can complain about my own kind. What's wrong with taxonomists? 死板板. What it means? They're only interested to see the animals for what they are, and they don't know how to use the information. These days, when I train my students, I tell them, when you discover a lot of interesting fish, when you give the data to a modeler to do planning, make sure you are part of that paper. And they tell me, boss, why? No need what? I done already. Give it to them, let them use. I say, you stupid. You are the one who created the information. You have every right to share with them when they write their big paper. That's how you should proceed. We cannot be so naive, so generous, because the world is not so kind. I will end by just saying that taxonomy is one of those fields in science, in biology, which is usually underappreciated. But it's one of those fields that allows us to find out what are the animals on this planet that we are sharing our planet with. You may not know there are so many species out there. You may not know there are so many wonderful animals that still need to be found. But when you see them, they're always lovely or cute, and it's important. And when you do conservation, when you do management, you have to know what they are. This is like basic science. You need the basic information to move to the next level. But please appreciate all those poor guys getting the data in the first place, because it's no fun. The best comparison is this. This is a discussion I had with a friend of mine from the army. You know, when you remember all the big wars we've had, in the last Second World War, when Japan was defeated, who was the hero? MacArthur. MacArthur saved Asia from the Japanese. Oh, really? And who are all those poor people who died out there, who you do not know the names? MacArthur was just the figurehead. All the other people have to do the donkey work. Well, taxonomists are a lot of these donkeys. That's why I like donkeys a lot. Thank you very much.
Let's again express our gratitude to Professor Ng for his wonderful lecture with a round of applause. May we invite Professor Chu to hold the Q&A section. Professor Ng, please. Professor Chu, please. And please feel free to ask any questions.
Okay, thank you, Peter. So, Peter always makes complaints. Okay, so are there any questions for Professor Ng? Well, there's a student going. Okay.
Hello, Professor. So, two questions really quickly. One is a bit more personal. What is the most memorable species that you have actually discovered on your own? And also another question, what if, let's just say one day I'm on the beach and I saw a new crab that I have never seen before, and being not really a professional taxonomist, how do I know if this is actually a new species or if this is actually a species that have been discovered by others? Thank you.
Okay, all right. First question is hard. Second one is actually easy. What is the most memorable one? I have actually no idea. All right. All the crabs I've found over the years have always been fun, because every time I find something new, it gives you a good feeling, because it's seeing something nobody else has seen before. I have no idea. Not sure which is the, he's asking a question like, you have 3 children, which is your favorite son or daughter? I like all of them, okay? Second question is easier to answer. You find a strange crab on the beach. You look at the books don't tell you. You look at the internet doesn't tell you, all right? Then if you're in Hong Kong, all right, you go to the universities and ask the professor on marine biology, right? Ask them, what is the crab? They will check. If they cannot identify the crab, very good chance they end up on my table. Because they'll say, oh, okay, who's the crab expert? Okay, Asia, Wan Peter, okay? And you send me a photograph. And some of you have done this before. And I say, never seen this before. Has it been preserved? And I hope the student or the professor say, yes, it has been preserved. Then my next question is, show me the animal. Then I'll check. After I've checked, I'll write back to the professor and say, yes, based on this information I've checked, it's not A, it's not B, it's not C. It may be new. All right? Now, before I make you too happy, all right, 99% of the times I get questions like that. It's not new. It's just a crab that few people have seen. Right? You remember, 200 years of history means almost all the common things have been found before. Right? Does it mean you can't find one? No, you can still find new ones, all right? I mean, I just named the crab with Stefano from, was it last year, Stefano? Yeah. So they go running around your mangroves in Hong Kong and find this tiny crab, all right? And for some reason, people have never seen it before. Lo and behold, surprise, it's a new species, right? Are there still more new species to be found in Hong Kong? Guarantee there will be, right? In time to come, there will be young people in Hong Kong, hopefully, that will learn the skills and take on the knowledge that I suppose the older people have and carry on to the next generation. The thing about taxonomy is that we always have to pass on this information one generation to another. The good news for Asia is this, and I can say this. 20 years ago, almost all the taxonomic experts are in the West. If you want to identify most animals, you look for Western scientists. These days, more and more is in Asia, right? Which is about correct. I'm biased, of course.
Another question?
You can ask me in Cantonese as well if you want to. Okay, that's a good question. I've asked that before. I've been asked, is it easier instead of having to spend all the time for the professors to go out and catch the crabs and look for the crabs, excuse me, to ask people to catch for you? The answer is yes. But there are 2 or 3 problems with that. Firstly is that, well, how much do you pay, right? And second thing is that the people catching the crabs, they may not know exactly what you want them to catch, right? So quite often, they may not get you exactly what you need. And as I said, there are actually a lot of species out there still to be found, right? So I can use that method if I'm looking for one particular animal, because I'm not sure what it is. If I know where it's found, what kind of place it lives in, I can ask somebody to get it, and I can pay for it. I've done it before. Okay. Question is that can all the information be put into a digital media like an app, and then from the app you decide if it's a new species? That has been discussed. People say can you just use photos? That's a problem. Many animals have the same problem. Plants also. If you give me a crab, you just look at the shell. I can't tell you if it's new. What do I have to do? Usually I have to dissect it. Well, for crabs, I take out the reproductive parts inside. I need to look at all the parts of the crab to be sure that it's new. And then because it's the science, that body has to be preserved. Why does it have to be preserved? Because maybe Professor Chu thinks my interpretation is wrong. He wants to check it again. That body must be there for the scientists to check. This is how science works. Somebody else must be able to check the evidence. Okay? So I need a dead body. He may decide, I know he does that, he may need the DNA to test it out. The body has to be there for him to use. So having a specimen on hand is very important because this is the objectivity of science. We have cases in science where people have beautiful photographs and they're all Photoshopped. And sometimes I can't tell. So for me, I know some people, for example, they have named insects, they have named animals from photographs. I do not do it because I prefer to have a body in front of me which other scientists can test in the future because that is the scientific method. And it's very important to have that body, those specimens examined because it allows you to see the whole picture. And that's why a simple application or digital media is usually not enough, right? It is not scientific enough as far as people like myself are concerned, right? And these days, in fact, to do taxonomy, there's another problem, right? And the problem is laws. More and more countries have very strict laws about what you can or can't do. And it makes it more difficult to study, right? So very often, to get the specimens, you have to go to the countries and you have to talk to the scientists involved, and it actually slows down everything. Using photos actually makes life easy sometimes. I've seen new species, people send me a picture, oh, I look at this, I know it's new. Then I have to contain my enthusiasm. I say, all right, catch me this animal, fill in all the legal forms, make sure you get the permits, and then I look at it. Then the process has to be done. The describing, the naming of an animal is not that easy. It takes time and effort, all right? But this time and effort ensures that the quality of the science is good. For the scientists here, you know, you write a paper, it needs to be read by your peers, refereed. Now, if I'm a referee, and I've been refereed many times, if the paper only has a photo, I reject. Why? How do I know this is not fake? I have got friends who actually publish papers with photographs given to them by very reliable people. The photos were fake. Their reputation, say fall, right? And it's very bad, okay? So I think for scientists, it's important to preserve your reputation because that's the most important thing we have, right? So the integrity of the science, no compromise. We need a dead body. But what you say about apps and technology is a very useful aid. If you have an app, I mean, we design apps for students. Common crabs on the seashore, you can use the app, helicopter, easy. But when it doesn't work, it means you may have something different. And that's where the scientist comes in, right? So you need to moderate the approach.
Maybe I have a question, Peter. Back to your very first question, okay? So now we have 2 million species that are named, but we believe there are maybe 10, 30 millions. So do you think that we can name them all?
Prof. Chu, of course, old friend, always must ask me the difficult questions. If there are 10 million out there and we only have 1.82 million named, will we fail? Yes, we will fail. We will not be able to name all of them because there are not enough taxonomists and not enough scientists. I'll be very blunt. And the world is a very big place. What are we to do then? Give up? Cannot be done, so let's give up. Cannot give up, all right? We have to keep trying to describe, to find as many as possible. We also have, therefore, to prioritize. How do we prioritize? Well, in Singapore, for example, or in parts of Southeast Asia, which I know better, if I know one particular forest is going to be destroyed, I prioritize. I go in there and try to find out first. If I know this place is 100% protected, 慢慢嚟啦，冇相干. Right? So, for taxonomists, we'll have to prioritize what we can do. There are too many animals, there are too few of us, but all we can do is try to get there as soon as possible, as fast as possible. We have no choice, right? Because the option is don't do. And don't do means you stand the risk of having a lot of species disappear without anybody ever knowing it was different. And to me, that's tragic. There are so many species. See, one of the things that taxonomy, I get very, very angry when I talk to many of my colleagues. Oh, we should spend another $1 billion to go to Mars. We should spend $1 billion to go to the moon. I say, why you want to go to the moon? You should go in the deep sea or the high jungle. There's so many things you don't know about your planet. No, they're not important. The moon is more important. I said the moon is dead. There's nothing on the moon that's alive. Or we need to collect a few rocks back. One of the tragedies for us as a species, as human beings, is this. We know more about the stars than we know about our own planet. The amount of money and resources we spend to study things far, far away is much, 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 much less than the money we spend to understand our own planet. And this is the only planet we have. Now that is very, very sad, right? I always think there's something wrong with the human brain. But that's another story.
Okay. Any more questions from the students?
I have a question, and that would be, when you go to the field and you make a rough estimate of what is new and what is not new, but then you may not get a very precise or accurate description of those, and you wouldn't get precise results unless you bring them into the lab and dissect them and then do DNA tests. So if you have a limited capacity of how many samples you can bring back to the lab, how do you decide which to bring and which to not bring? Thank you.
Okay, good question, tough question. Well, if you ask me, I'll bring the crabs back and I'll leave everything else behind, right? Because I'm biased. Now, taxonomists work the same way. We are very passionate about the kinds of animals that we are expert on. When we go to the field, we make collections, we make choices, right? And you're right, we make estimates, and we bring back what we can, okay? And we bring back only the things that we can handle. And that is not always the complete picture. I agree. The picture is not perfect and is incomplete and we are biased, right? But we don't really have a choice because we only have so many people and so much resources to do it. So sometimes we only have a small snapshot of the whole picture. What do we do with the rest of the animals? Well, we don't throw them away. We preserve them, we pass them on, put them in a museum, or pass them on to somebody who wants them. For me, when I go to the jungles or go to the deep sea, the only animals that I will catch and preserve and bring back are those for which I'm studying or I know my colleagues are studying. I do not catch those for which nobody is working on because it's a waste of killing them and waste of my time, right? Are those choices correct? Sometimes I wonder myself, right? But I don't really have a choice. So we have to make choices. My argument is simple. Better I know a bit than I don't know anything at all. Yes, to start somewhere.
Our head has a question.
Yadin. Yeah. Mike.
Well, this question actually haunted me for a long time. I think Professor Zhu knows what the question is. Now, some months ago, Professor Zhu, the chairman, gave a short talk in Inno Carnival. I think the name of this occasion is Inno Carnival, and Professor Lep Chi Chui was the moderator. And the question from the audience is why crabs walk sideward? And 打橫行, okay? And the answer, actually, Lep Chi Chui tried to answer the question. He said crabs learn from Homo sapien, learn from a human being. And, well, I always want to know what is the answer. Please.
That is the most interesting question of the night. Why do crabs walk sideways? Actually, to answer that question, crabs don't have to walk sideways. Many crabs actually can walk forwards and backwards quite efficiently, all right? But because of the structure of their body, most of the crabs, that you see in the market, okay? Because their body is, rectangular, their legs are on the side. The easiest way to move is left and right. Can they move forward? Yes, they can, but slowly. Move sideways for many of them is faster. But if you go to see the crabs in the sandy areas, some of the ghost crabs and the more round crabs, they can move front, forwards very fast, right? So not all crabs move sideways. The crabs that I work on, the 7,000 over species, they actually move around in almost every direction you can think of. Some even swim. So there's a lot of ways they move. So this idea that crabs only can move sideways is a myth, not real. And as to that part of the answer somebody gave is learn from human beings, that is absolutely not true. Crabs are much smarter than to learn from human beings. All right? They have been doing this a bit longer than we have been around. So, very sure they didn't learn from us.
Okay, we have time for one last question, if there is any. Okay, so if there are no more questions, we'll call this to an end. Thank you. Thank you, Peter.
Once again, thank you, Professor Ng, and thank you, Professor Zhu. Now, Asia College would like to thank Professor Ng for the lecture. May we invite our college head, Professor Harry Wong, and Professor Wong Kam-po, Director of School of Life Sciences, to present the souvenirs to Professor Peter Ng. Thank you, Professor Wong Kam-po, Professor Harry Wong, Professor Zhu, and Professor Ng.